Okay, so now I'm going to talk about the pupil systems, and I'm going to post this. I'm going to talk a little bit first about some systems in the pupil stage that describe the cell dynamics observed during the cyclone rotation. And this is the paper that looks much similar that is found in the back of the fly. I will talk a little bit about that. Also, I will talk about other papers that they have been talking about the same behaviors, the same cellular process that we described, and that they are related to cancer. And they also have been described in the back of the fly that is called the nodum. Yeah. So let's start. So there are two papers that they have been talking about this type of cell extrusion due to crowding. So so those papers appear in 2013, and one combined results from zebrafish that they did live imaging, human colon, and these in vitro cells. So they grow cells, and the other one was in the fly nodum. These two papers appear at the same time, and they talk about something similar. That is when there is compression. So here, for example, during development, there is compression in the nodum. This part of the tissue is moving in this direction. The two wings are getting the two tissues that surround the wings are pushing, are moving, and all these cells in red start dying because there is compression. And something similar happened here. They have this machine that they mimic the process of compression, and they found that cells died also because when there is compression, the cell density increase and there is crowding. So same thing they said that happened here in the fin of the zebrafish. So now I'm going to focus on this process of the fly nodum, and the idea that simple physical forces play a key role regulating cell density, and there's this fundamental process. This is a fundamental process to understand early stages of cancer, so the transition between a normal epithelial to a cancerous epithelium. So, and this is what I just said, and that is very important that most of cancer have an epithelial origin. When you hear about carcinomas, carcinomas are cancers that have an epithelial origin, and most of the cancers have an epithelial origin, so they go from a normal epithelium to a to an epithelium that you start seeing tumors everywhere, that they spread everywhere. However, we don't know exactly the transition, how you go from a normal one to one that have pieces of tumor everywhere. And to answer that question, we know that this is a very important process, the balance between proliferation and cell death. And here you have a uniform epithelium. And what happened during development is that you need to repair that epithelium. So during lifetime, to repair their epithelium, you cannot leave holes in the epithelium. You cannot kill a cell and leave a hole, or kill all this part of cells. Yeah. So how they do it? It seems that what happened is that there is proliferation. So cells start dividing. Then when you have too many cells, what happened is that now there is less proliferation and more death. So, for example, all these regions in the red, all the cells in the red region are going to have more cell death, and in that way, some of them will die. And it has been shown in that paper that this process happened in a relatively stochastic way. So, in every single cell, in every single fly, it will occur in a slightly different way, in a similar way to what it happened in the sexcom. So just local events are responsible to determine which cell died and which cell doesn't die. Uh, they said that this was a crowding-induced cell extrusion, and this process of crowding cell extrusion seems to be very important for cancer. So they found something very similar to what we saw in the proximal region of the sexcom, that when there is crowding, there is an increase in cell density, cells start dying. And what other papers, and I'm going to talk about that in a second, is that this seems to also happen when in a cancerous epithelium. So what we are going to in the next following papers, what they did is let's modify, let's use fruit flies that have with genes that we found in human cancers and see what happened. So what they did is, okay, let's create pieces of tissue that have these genes. So it's a genetic perturbation, uh, that you create a little tumor. So they have done this with genes like BRAS-B12. That is very important. It has been found in 30% of cancers in humans. So when you have a mutation in BRAS, there is a problem. They have found this in other ones like MYC, that is also found in cancer, and you will see a lot of genetic perturbations. And why they don't turn the entire tissue into, or the entire fly into, they don't use mutations that all the cells have a problem, a genetic perturbation in one of those genes. Why? Because the flies are going to die right away, or you cannot see how cancer progress. So you have to create a clone. You have to create just a group of cells and see how they compete with the other cells. So what they found is that, and I will show you several examples, is that around the tumor the cells will start dying. Yeah, and it seems that is due to there are multiple reasons. One of them, in some cases, is because there is more compression as the clone is going to start increasing in size. There will be less space for the other cells, and the cells are going to do exactly what happened during the sexon rotation. They will mix, they will die, and some of them will start getting smaller. So, um, so the following papers start. I'm going to show you the information, and most important, I'm going to leave the article for you to give a quick read. So this paper, um, what they did, they follow the work in the NODM, and they correct some problems that the previous article had, some mistakes. And what they did is said, it's true. When you have a wild type, and this is the wild type here, you have wild type cells, and we found that there is a space, a protein that is involved in the cell death. So you find that as there is compression in this region in the middle, cells will die. Yeah. Now, when we create tumors, when we have genetic perturbations with RAS-B12, the cells, what is going to happen is that they are going to start growing, and around the cone, what is going to happen is that the cells is going to start dying because they have less space, and they start going to get smaller. Okay, so they found that there's tissue crowding, then the space activate, then the cell dies, and in that way they release space. So this is what happened in normal cases, and this is what happened in cancer cells. So in other words, in these cases, in the RAS-B12 case, the tumor cells, or the genetic perturbation, uh, outcompete the wild type, and they start killing as the tumor grows, start killing the wild type cells. So you can give a look at this paper, and this is the main idea that is very important. And one section of that paper that I think is important is the cell size. So what they found is they measure in this graph the cell area. So they compare the cell area, and when you have here, this is the control. This is a control when there is no effect. Yeah. So here is a wild type, and when you have the clones, the cells can get bigger. However, when you have the clones with the tumor, the cells get much smaller. Yeah. So much smaller, closer to the com. So this is sorry, closer to the tumor. So here, what they found is every time that you have a clone, the cells are going to start getting smaller. And this is something that it seems similar to what we found during the rotation. So if you remember, every time that we're closer to the com, something different is happening. As the com cannot die, as the com is like a barrier, similar to a tumor, they both don't die. You find that close to the com, the cells start getting smaller. If you remember this area, however, if you get more to this area, the cells can get are bigger, and so this is a parallel that we can talk that we are using this system to study cancer, and in some way the com is like a tumor that is growing and is releasing is giving as it rotates is moving and moving. Yeah, the space above the sexcom, the cells are getting smaller and smaller. So in the beginning you have all this space, later you have less space, later you have less space. So how do you solve this problem of having less space? In both cases, both in the tumor or in the wild type, you use the same mechanism. That is, you kill cells, you make them smaller, and you mix them. Yeah, and even here we find something similar in your research. However, nobody that I know has studied in a lot of detail, like this thing that you did, the time and the area. I don't think that anybody has studied how the cells get bigger or smaller. They just study the beginning and the end. So I don't think that anybody has studied these oscillations, and I think this could be something interesting that you can talk for the future projects. Okay, so now in this paper, they also use the node on the back of the fly, but in this type, in this case, they focus on the cell mixing, and instead of using RAS, they use another gene called MEC. And here they create a different genetic perturbation. Here in green, you will see the flies, the winner cells, yeah, the cells that are going to win in this competition between the normal and the wild type. Sorry, between the the mutant and the wild types. So you have winners and losers. Sometimes the wild type is the one who wins. Sometimes the mutant is the one who wins. In this case, they pick this, and what is interesting here, they label it here in purple, the wild type, if you see, and in green you will see the mixed cells. So they create that, and what they found out is that if you see during when these cells that they label here, they start mixing. Yeah, they start separating. They have a lot of reorganizations or cell rearrangement. So similar to what we found in of these stochastic movements, when you have a tumor and you have cancer around the cells that reproduce this cancer environment, you will find a lot of cell mixing. And they they found that this mixing can be due to two reasons. So in some cases, it could be because the cells are dying, yeah, and in some reasons just because the cells are mixing because there is junction remodeling. So if you remember during the SAXCOM, here there is cell death, and here the cells are mixing without cell death. Yeah, they're in the region 1. I think this is the region 2. So this one resembles this, while this one resembles the cell rearrangement caused by the cell death. So it's very interesting because still we can find some parallels. Then something that we haven't done here, they talk more in detail about what happened with the cytoskeleton and how the cells decide which cell will die and which cells don't die. And they found that that is based on the actin. When you have this structure here, if you see, the cell boundaries have actin, so they describe in a lot of detail how this can happen. Um, but this we don't have to. You don't have to read these sections. You just have to remember that again there is a parallel to what happened in the sexcom and what happened here during the early stages of cancer that we have been trying to mimic using fruit flies. So here there are articles that that are discussing this relationship because between mechanical tension and cell death, but this one, instead of being in fruit flies, that are in our organisms. So here is the one that I already discussed about the fin fish, and about the cell culture. Here is a review that talk about this point, and here you have more examples of this issue that is just growing and growing, and it seems to be very interesting. Here you find a lecture of this lady that talk about how actin, you find this actin region, actin outline, active ring, sorry, here they said, and this active ring is the one that allow the cells to die, and she talked about this cell extrusion, and yeah, I don't know if we will have time to watch it, but it's something similar to, it seems that it's a process that happened in different organisms and also in flies. And here that is related to this topic, that is cell competition, that in the sexcom or in cancer the cell seems to be competing, and so here you have in blue here the wild type cells, and here you have a less fit cell. So what is going to happen is that the less fit cell is going to die. So here you find a case when the cells are competing and some of them will survive. What happens when you have a super fit cell like the RAS-B12 that we described is that those cells, so for example a early tumor will start killing the cells around it, and as there is more space they can start also proliferating. And this topic is also growing. It's a growing field. It started a long time ago, like 20 or 30 years, but now with live imaging it seems that it's coming back. So for example, there are different type of competition that they are trophic interactions. There is competition for space and fitness sensing. Here the one that it seems to be related to us is this competition for space. So I just gave you some topics that you can discuss, and I think this topic of cell competition, it could be something that you can discuss in the paper. Yeah, I just saying that the process of rotation, yeah, mimic cases of cell competition, that they have been seems to be important for cancer progression. Yeah, and so for example, topics like that close to the regions, the cells behave have a similar behavior to those found close to a tumor. Yeah, if you remember that, that is important. Here, suppose that there is a tumor and that there is mixing, that there is a lot of behaviors. I don't think that you should write 2 pages of that, just 1 or 2 sentences. And also said that future studies uh, should work more on that, and in particular, we don't know what happened with the oscillations. So we don't know if there is oscillations in tumors, or maybe you already found an article that discussed that. But the importance of our paper is that we are describing how those oscillations can take place, and that is something that it will be interesting. And if you have more questions, just let me know. And these are just giving you some topics for discussion.